My name is Maya Kotas, and I am a postdoctoral fellow at UCSF. I'm here today to tell you about my project on Sentinels of the airway, discovering the role of sensory tuft cells in airway inflammation. Long before I became a pulmonologist, I knew that breathing is a fundamental requirement for mammalian life. But while the air we breathe provides essential oxygen, it also presents a constant threat since it is full of microbes, particles, and chemicals. These days, as a result of the COVID-19 pandemic, we are all more aware than ever before of how dangerous it can be to just breathe. Our airway, the passage that leads from our nose and mouth all the way to our lungs, protects us from most of these threats. To do so, the cells lining the airway are precisely maintained and in close contact with the nervous and immune systems. Mucus cells trap bacteria and dust so that they can be swept away. Sensory cells interpret inhaled chemicals to differentiate between things that we need, such as food or mates, and those that may harm us. And immune cells survey and control the balance of healthy and harmful microbes. When these processes fail, diseases such as pneumonia, bronchitis, nasal polyps, and asthma can result. If we could elucidate the intricate functions of the healthy airway, perhaps we could prevent or treat these conditions. To gradually learn how these many specialized cells in the airway protect us from the outside world, I am focusing on one specific type of cell, the tuft cell. Tuft cells, so called because they have a tuft of tiny sensory feelers reaching into the airways, are also found in the lining of the intestine and in the taste buds on the tongue. We think that they taste molecules that land on their surface and then secrete signals to activate nearby nerves and immune cells. On the tongue, those signals help us interpret whether foods are pleasant or spoiled, while in the intestine, they activate immune cells to make IL-13, a molecule that causes tuft and mucus cells to sweep the insulting chemical out of the intestine. Through these mechanisms, tuft cells are critically important in sensing and protecting us from the outside world. But what are tuft cells doing in the airway? We have some hypotheses. You see, the cells and molecules in the intestine that are activated by tuft cells are all part of an arm of the immune system called type 2 or allergic inflammation. And those are the same types of cells and molecules that are activated during airway allergy and cause diseases like hay fever, asthma, and nasal polyps. Those allergic airway diseases are common and debilitating, affecting 10 to 20% of people, sometimes with repeated life-threatening attacks. There are few available treatments for these diseases, and no therapies that are effective in all patients. I think that tuft cells in the airway may have a role in activating this type of inflammation, just as they do in the intestine. Perhaps they do so in a misguided attempt to protect us from something that we breathe. In order to understand how tuft cells interact with other cell types in the airway, and how they contribute to protection and pathology, I'm using a two-pronged approach, examining tissue from patients with nasal polyps and asthma, and in parallel, creating models to study these cells in mice. To understand the role of these cells in human disease, I'm collecting cells brushed from the surface of human nasal polyps or healthy sinus tissue. Having sampled those cells, I'm using a technique called single-cell sequencing to measure the gene expression in each individual cell and determine whether Tuft cells are different in the sinuses of patients with allergic airway disease. Through this work, I found that nasal polyps have an increased number of Tuft cells, and that those Tuft cells have a different gene program from the one in healthy patients. But to move beyond observation and investigate how and why these cells might change in patients, I'm using mouse models. Using genetic engineering, our lab and others have developed mice with Tuft cells that are fluorescently labeled or that have specific genes mutated. These fluorescent labels, or reporters, allow us to actually see Tuft cells as they grow and interact with other cells in the airway. Using those reporter mice, I've seen Tuft cells in two different tissues in the nose, the nasal epithelium and the olfactory epithelium, as well as in the larynx and trachea. I've observed that they are more frequent in some of those tissues than others, grow at different rates, and have different genes that they express. Intriguingly, in the nasal epithelium, the portion of our nose that is designed to protect against particles and organisms, I've seen that Tuft cells increase in mice with allergic inflammation, just as they do in patients with polyps. By contrast, in the olfactory epithelium, whose main purpose is our sense of smell, Tuft cells are unaffected by allergic inflammation. Perhaps this means that Tuft cells in the olfactory epithelium are more like the ones on the tongue, interacting with nerves to sense the things that we taste and smell. And maybe the Tuft cells in the nasal epithelium are more like the ones in the intestine, which orchestrate allergic type 2 inflammation. By learning how Tuft cells choose to interact with nerves or specific immune cells, I hope to manipulate these interactions and find ways to protect patients from allergic airway diseases. There are not enough treatments to help patients who suffer from diseases like asthma and nasal polyps, largely because we don't yet understand the mechanisms of these diseases. The studies I've described will identify lines of communication between Tuft cells and other airway cells and help teach us how the airway protects itself and how this sometimes goes awry. While these studies are just a start, understanding these communications is an essential step towards identifying new targets for therapy. As a physician and as a scientist, I'm committed to unraveling these mechanisms, investigating the role of each of these specialized cells in the airway to help us breathe easier, to help us find new cures.